Well, thank you very much for inviting me. It's a great pleasure to be here, and I thank the organizers, and I thank them for, I thank them very much for, it's all right? It's here? Yeah. So I thank you very much. It was fascinating for me, and to some extent I will sort of consider some of the things that have been said here already and add to them a little bit, and we can discuss it. So I also want to thank my friend Anna Zeligovsky, who did the pictures, and you will see some more of her pictures here. I think that's the only thing that you probably remember from this lecture, but that's very good. So I thank her, and this lecture is dedicated to the person with whom I worked for many, many years, my best friend, Marion Lamb, who unfortunately died in 2021. So let's go back to Mendel's paper and to the background from which Mendel came. As already Greg said, in 1837, the Agricultural Society Journal was discussing the 1836 meeting of the Sheep Breeding Association. And this meeting was discussed by the participant, and among the participants was Abbot Cyril Knapp, who summarized the discussion on the problem of heredity. And he said, and this I'm quoting Orell, his book from whom I took this quotation, from his book of 1996. He, Knapp, emphasized that it was not only the question of breeding process, but the question to be answered was what is inherited and how. And this was something that Knapp asked. Knapp was an intellectual. And I want to say, so was Mendel.
Recording in progress.
Right. Thank you. And this was a very important question. What is inherited and how? And it could be asked within all kinds of contexts, within an evolutionary context, which required some form of heredity for it to happen at all, as Darwin well realized and as Mendel well realized, and also, of course, in the context of breeding and of crossing varieties, which was extremely important for agriculture. And in Moravia and in Brno was a center where these things were discussed, and Moravia was a place where a lot of controlled breeding was taking place. So I taught Mendelian genetics since, I think, the Dark Ages. Well, I'll tell you, 1987. And in order to teach it, I read at the time Mendel's paper. And I based, in order to explain the historical origins, because I thought this was interesting. Just to teach Mendel's laws it's very easy, so to speak. But to understand how people came to think, how he came to think about it, that was complicated, and that was interesting for my students, who were very, very good students, I must say, because I was teaching the excellence program in Tel Aviv University. So I had to interest them. So I took Mendel's paper, and when we were going through the explanation, I talked about the paper, and I talked about how we see Mendel now and and what Mendel actually said in this paper. Of course, my reading of the paper was not unbiased. And what I'm telling you now is also not unbiased, of course. I taught the paper because I taught the paper for many reasons. One was because it was kind of model of clarity. If you want to teach the scientific method, you take Mendel's paper. This is the context of justification at its best, as we say in philosophy of science. This is how you look at hypotheses, you test them, how you choose, how you do the statistics, what is your sample size, what you take into consideration. It is a brilliant paper. So that was one of the things. And the other things were the kind of regularities he observed, what he called laws, or what we call laws. And usually it is presented like there were three laws: the law of segregation, the law of independent assortment, which of course we know depends on linkage. He chose very carefully his samples, his traits. He started with 22, he ended up with 7. He didn't tell us exactly how he came to this 7, but they are unlinked. And this cannot be a coincidence. And dominance, there is no such law, by the way, in his work, that there is no law of dominance that he's talking about. This is something that you find in the textbook, but I didn't see it in the paper. That's very interesting, although dominance is very important for him because he wants to look at some kind of regularities of segregation, and this is what he wants to study. This is how he wants to understand what is going on during crossing. And this makes it easier for him, although he realizes, and he says it in the paper, that there is polygenic inheritance because he saw it in the beans. He looked at, he saw that the color of the bean cannot be explained by simple dominant recessive characteristic in one locus, as we would say today, just one factor. He has to take into account several factors, and he went even beyond that in a few sentences here and there. But the most important thing for him, and the most important thing that I tried to teach my student through his paper, was let us look very, very carefully at the assumptions underlying the developmental law for, most important developmental law for him, which was the law of segregation. So the first thing that is very striking about it is he wanted to find some constancy, some regularity, and in order for that to be possible, he has to look at variation. So constancy and variation, the variation, of course, has to be of a certain type. It has to be stable, for example. The variation itself has to be stable in inheritance. But nevertheless, it is the inheritance of variations that will explain some kind of generalities about crossing, about the behavior of hybrids and their progeny. So, and he's, of course, looking at these general regularities in sexually reproducing organisms. Now, what is he assuming? What are the assumptions that underlie, and he is aware of them underlying, his experiments? The parental origin is unimportant. He stresses it several times in his paper. It doesn't matter if the dominant trait is in the mother cell or in the sperm. Really doesn't matter. There is equal segregation of parental factor types of the elements. So there is regularity that it's not as if one of them suddenly dies. Yeah, they are segregated. There is no effect of dominance of recessive nature of one factor on the other factor in the reproductive cell because segregation. So they are not contaminating each other. For example, if you have two factors, one dominant, one recessive, two elements, the dominant one is not changing the recessive one, and the recessive one is not changing the dominant one. This is an assumption. Otherwise, it wouldn't work. There are no hereditary influences of the condition of life on the hereditary stability of the elements, on the hereditary stability. He's very well aware that the conditions of life can change the appearance, but not the hereditary stability, because he said that even after many generations, you can see the factors in the right environment. He's very aware of the effect of the environment on development, but not on heredity, he says. So this is something that is already in the paper. You can find these assumptions in his paper. So I said, look, let us look at these assumptions. They are very interesting assumptions. And they are necessary for understanding his results, and not only the results on peas, because he was experimenting, altogether on 16 species, different species. Not always did he find such nice results as he's found with Pisum, but he found very nice results. Okay, so let's look at this, I said to my students. Parental origin unimportant. Is it true? Very often it is true, but we know, and I'm talking about 1987. We already know quite a lot, and I know quite a lot about imprinting because it is my business to know about it, because I'm interested in epigenetics, right? So, so we know about imprinting, where the origin of the cell, whether it's from the pollen cell or the egg cell, matters to the expression of the trait. This is what imprinting means. You don't change the DNA sequence, but the expression is different according to the parental origin. We know about mitochondrial inheritance, which is only maternal, and we know about maternal effects. Now, these things suggest that the parental origin is important in some cases. How many cases, when, how much, we'll leave it. But we know that this generalization of Mendel is breached for different reasons in many cases. And we know also that already Correns, who worked, one of the rediscoverers of Mendel, was noting non-Mendelian inheritance, which we know now is chloroplast inheritance in hybrids. Now, equal segregation, is it correct? Very often, yes. But of course, we have abnormal cases of non-disjunction, which wouldn't worry Mendel because abnormalities happen. But we also have something called meiotic drive, where one of the products of the segregation leads to the death or poisons the other gamete, leads to the death of one of the gametes, right? So we already know about meiotic drive. I'm talking again, 1987. Of course, Mendel didn't know about any of these things, nor do, but what I'm talking about are these assumptions of his that we're looking at them one after the other, and I'm looking at them because I want my students to realize the kind of what he discovered, and also the exceptions to this, and some exceptions are not very exceptional, actually, right? Now, no effect of the dominant or recessive nature of the element on the other element, right? There is no contamination between the elements in the reproductive cells before segregation. He is aware of the fact that there is a possibility of generation of new stable products of interaction in reproductive cells, because we're talking about constant hybrids, and Greg already talked about that. So he's aware of constant hybrid, but he's interested in the non-constant one, the one that are segregating. And in those that are segregating, he said that there is no effect on the recessive or dominant nature of the element, in those that are segregating. Although he is aware of the fact, by the way, which is also interesting, that you have hybrid vigor. He says this in the paper, that sometimes they are luxuriant, as he puts it. And he also says that sometimes they have extreme traits that are not seen in either the dominant or the recessive parent. He is aware of that too, that they are extreme to, for example, he's talking, I think, about seed coat, and he's talking about the color of the seed coat, and it sometimes is kind of patchy, has black blots on it or something like this, and he's saying this we don't see in either in the dominant. We see a little bit of that, very, very little. Now, what do we know already then? We have gene conversion. We have phenomena like gene conversion, where one of the genes, he of course didn't know about genes, I'm talking about what we know about in 1987, one of the genes converts the other genes to its own image, right? So you have a recessive and a dominant, or A1 and A2, and A1 is converting A2 to become A1. Now, this is, you can say this is rare, depends where. In the East, for example, it's not very, there are strains where it's quite common, gene conversion. And we know already in 1987 about paramutations, because a plant biologist called Brink talked about it, where this is something like gene conversion, but unlike gene conversion, it doesn't behave as stably as a genetic variation. We know now that it is epigenetic, right? It's basically epigenetic gene conversion. This is what paramutation is. And we know about it already because Brink talked about it. Okay. And there is no hereditary influence of the condition of life on the stability of the hereditary elements. Now, this was quite important for me for obvious reasons, because this is what I devoted quite a lot of my working life to, was to look at this aspect of inheritance, look at the effect that the conditions of life, the development has on the hereditary stability and on the hereditary nature of the element, whatever it is. And if we're talking 87, this is when Marian and I are beginning to think that maybe that we're trying to publish a paper unsuccessfully about epigenetic inheritance. Our first paper was published in 89 after rejections by Nature and Science for having no interest for the general public. And thanks to John Maynard Smith, who read the paper and said, well, that's interesting. You have to publish it. And he wrote to Wolpert, who then said, okay, I'll publish this stuff. And that was it. So thank you very much, John. But anyway, so I was very interested in soft inheritance. And I was interested in, and we knew already that there's stuff in the literature that is best explained with the assumption that the conditions of life do change the hereditary nature of whatever is transmitted. Now, it's not that there are at the time we knew that there are alternative explanations also. You can do, by the way, with a few genetic elements and some kind of interactions that you invent, you can explain every genetic trait that you like. I think it was Morgan who said that give me 6 genetic traits and I explain every result of breeding. You can. If you're a good geneticist, believe me, you can. What you can't do is predict the next results of the breeding experiments. This you can't do very easily. You can't do generally. Anyway, without a lot of auxiliary assumptions. So these are the assumptions. So here we have the assumptions, and what we know is going beyond the assumptions of Mendel. But it is very important to remember what Mendel's assumptions were and how he thought about it. And it's also very important, if we know about these exceptions for the assumptions, to expand our notion of biological heredity to accommodate these cases. And especially for my case, it is very important to include the possibility of soft inheritance, that inheritance is not hard, but that the hereditary nature of whatever is transmitted can be influenced. Not always is it influenced, but can be influenced by the conditions of life under some conditions, in some cases. It's very important to include this in our notion of inheritance. So I devoted my life to this. This is not, by the way, I'm not trying to promote myself, but I'm just trying to tell you that this is what I did for a long, long time. The first book was 95, the second book was 2005, and then 2014, and this one is 2020. So I was thinking about it with Marian a lot. All of the things that I'm talking about is something that we both discussed. And a lot of it was discussed, by the way, in the context of teaching, how to best teach heredity to intelligent students. How can we explain heredity? Because we were interested, and of course evolution, because we were both thinking evolutionarily, and this was the impetus and the motivation to explain evolutionary patterns, to explain how things can work out during phylogeny. So we thought we have to expand somehow the notion of what inheritance is. That inheritance is not just the transmission of genetic factors, and evolution is not just the change, as Dobzhansky said, of genetic factors in populations. Yes, this is part of the story, but it's not the whole story. that inheritance is in fact the change in heritably varying traits over evolution. Traits. And the inputs into the inheritance of trait can be different. There can be different inputs into the heredity of traits, and these things will interact almost certainly. Not necessarily, not in all conditions, depends on the time scale we choose, depends on many complex factors. But we don't have, if we want to explain something, like for example, the tendency of alcoholism to pass in families, how do we explain it? Is it just a genetic tendency? Maybe it is. Is it enough? No, it isn't. Is it also, what kind of cultural reconstruction, what kind of developmental niche construction goes on in these cases, and so on. So this picture is a picture, so it's, we thought of inheritance as a transmission of variations between generations that is based on some kind of evolved memory transfer mechanisms. Very general, deliberately very general, in order to accommodate genotypes and epigenotypes, social types that are inheritance to social learning, and in the human case, inheritance through the symbolic system, which is a very strong inheritance system in our terms. And what this picture shows is a kind of depiction of what she calls the 5 mothers, the kind of transmission of genetic factors through the egg cell. She's looking at females here, not because males are unimportant, they are very important, but just because of simplicity. She's looking at the non-DNA factors in the cytoplasm and on the nucleus, epigenetic factors of different types. She's looking at transmissible effects in utero. She's looking at early developmental effect, which can be reconstructed from one generation to the next, for example, food preferences. And she's looking at the gray picture of mother culture in humans, symbolic culture. So there are many, many inputs and many inroads into what makes a phenotype, not only what makes a phenotype developmentally, but what makes a heritable variation in phenotypes. And for that, I tried to talk about the not very elegant term, heritably varying traits, HVT. The HVT is a heritable, this is what we should look at when we are looking at evolution and inheritance. We should look at HVT and then look at the factors that contribute to the hereditary stability of this, or instability, or modification of these traits. That's, we thought, how we should think about heredity and evolution. Looking at traits, going back but the trait, of course, you can sometimes say, well, one of the factors that I'm interested in is a genotypic factor. I want to put all the other factors into the background to control them as much as I can. Just look at this. That's fine. You can do this. You should do this. We have to do analysis, not only synthesis. So, but this is the way that we were thinking about it. Tell me how long I have, by the way, because I forget myself. So the main work that we did was epigenetic work. We started with epigenetics. And we know about, so here we have genetically identical but epigenetically different individuals. At this, with the exception of the mice, these are not heritable variations. They are just developmental variations that are different. We have two identical twins, here and here. The two girls here, up and in the bottom. They are genetically identical, phenotypically very different in many, many aspects of their cognition, behavior, and so on. Here we have genetically identical as can be. Of course, nothing is totally identical in the world, and there are all kinds of genetic variations. But mostly, we can also assume that they are neutral, phenotypically, that they don't have phenotypic effects in many, many cases. Not always, by the way, but in many cases. And this kind of statistical work can be done and has been done. So we have here the bees, we have the queen bee, and we have the female workers genetically the same, phenotypically different, epigenetically very, very different. And the mice are very interesting because it's a pure line, and the difference between the two, the fat one is yellow, obese, has tendency to develop cancer and diabetes and whatever, not very healthy mouse. The other brown mouse is normal. Genetically, as far as we can tell, they are the same. Epigenetically, they are different in that a particular region is more methylated in one strain and less methylated in another, allowing the expression of a particular gene. And this expression of the gene has stable effects, and to some extent it is inherited. To some extent, not completely, right? Doesn't have the stability of genetic inheritance in this case. Now, what about the genetic stability of epigenetic variations? Varies a lot. Varies according to the taxon we are looking at, the group of organisms we are looking at. Depends also on the kind of sequence we are looking at. Depends also on the conditions in which this variation was induced and for how many generations it was induced. Many factors can affect the stability of epigenetic variations, but they exist. Not only do they exist, they're all over the place and in all organisms. Well, we didn't know that in 1987, but we guessed. Okay. So I'm talking about epigenetics, and I'll say that very briefly in this audience. I don't need to say epigenetics has a history. The term has changed. The original term is, the word was coined by Waddington, and it says, it's in the wide sense, epigenetic denotes the branch of biology which studies the causal interactions between genes and their products, which bring the phenotype into being. And it's this bit here. Yes, these are the genes. The pegs, the black pegs are the genes. Here you see the kind of macroscopic phenotype, and here you see the gene network. And the gene network, he called it the epigenotype. And he didn't think about epigenetic inheritance. I want to stress it, he didn't. When he was thinking about transgeneration and non-genetic inheritance, he was thinking mainly about culture. He did say in one sentence somewhere that there can be sometimes, but only very little, non-genetic inheritance. But that was it. So when he was thinking about epigenetics, he was thinking in this broad term. But what he was stressing, and what was very important, was the stability of this network. The stability of the epigenotype, which is highly canalized. Highly canalized means that that we will have the same phenotype in spite of different variations in both the environment and the genes. The network will sort of reorganize itself so that the macroscopic phenotype remains more or less stable on the one hand. On the other hand, this network has the possibility of plasticity, which means it can reorganize in ways that where the phenotype is different but adapted to the environment. Usually adapted, not always. Okay, so epigenetics in the narrow sense is the study of developmental processes that lead to self-maintaining changes in gene expression that underlies the states of the organism, their components, and sometimes their lineages. So in this term, this notion of epigenetics includes epigenetic inheritance. And epigenetic inheritance is the transmission between generations of cells or organisms of epigenetic variations. Okay? Now, what are epigenetic variations? I will not go into it, and I don't know how long I have. Okay. So epigenetic variation, there are different classes of epigenetic variations, and we begin to understand the biochemical mechanisms of some of them. When Marian and I started working, what was understood and what I did my PhD on was DNA methylation. I started being interested in epigenetics because my PhD was on X inactivation. In female mammals, you have two X chromosomes. And one of the X chromosomes is inactivated and stably inactivated in early development. It's either paternal or maternal chromosome in eutherian mammals like ourselves. In metatherian mammals, it's the paternal chromosome. And it's stable. Usually when unless it gets reactivated during meiosis. in the germ cells of the female. The inactive X chromosome gets reactivated, and then they segregate, and the mature egg cell, the egg cell has just one active X chromosome. Now, the question that was obvious, and what is the basis of this incredible stability? I mean, it's not just one gene that is being repressed or activated. It's the whole huge chromosome. What's going on? How does it happen? And why is it stable? In inheritance within a lineage, developmental inheritance. So that was, I was extremely intrigued by this question for all kinds of reasons. And one of the things that we know now a lot about it, about the mechanisms, I will not go into it, RNAs are involved. At the time we didn't know that. I looked at methylation, DNA methylation, because that was the only system, epigenetic system, that was known at the time. And I looked at it in order to see whether it is or isn't relevant to X inactivation. And in cell lines, it is relevant under certain conditions. Never mind. But, you know, it's a messy, messy, messy project. I almost left my PhD because of that. So anyway, but when we're thinking about epigenetic inheritance, we're thinking about different kinds of processes that can lead to the reconstruction of the expression of genes or the products of genes in the next generation of cells. Of cells. I'm not talking necessarily about meiotic cells. I'm talking first about mitotic cells. How is mitotic inheritance possible? This is the first question that I wanted to understand, and then meiotic inheritance, because it was clear that there must be some, that if there is mitotic inheritance of these things, it cannot be all wiped clean. There is no delete button in germ cells. There is, yes, a lot of it is, you have to reset it so that it is totipotent, that it can develop into the whole organism. But it can, but there is not just one totipotent variant, right? There must be multiple totipotent variants that carry some memory of what was happening. This was our assumption for pure biological reasoning, right? We said it's just impossible otherwise. That's not how biology works. And the first thing we did, so we looked at mechanisms, we looked at what was known at the time was mainly about DNA methylation. A little bit about histone modification was people were beginning to work it out. Very, very little about RNAs. That came later in the 1990s. There was self-sustaining loops, mainly in bacteria. They were studied only from the 1940s. And we looked at all these things. And now we know a lot about it, a lot more about it than we did. So this is what we were looking at when we were doing the epigenetic inheritance thing. And we were thinking about, 3 minutes, okay. So I won't talk about too much. So we were thinking about this broader, so we were thinking about how our variations, epigenetic variation, and we were also thinking already then about other types of variations that are phenotypic variations that are inherited from one generation to the next. And I talked about those that occur through social learning and those that occur through the symbolic system. And we thought more or less in these terms. This is not our diagram, but more or less. We thought about there can be transmission from germ cell to germ cell. This is number one. This is what Mendel was looking at. The germ cell is a developmental bottleneck, right, in inheritance in many, many cases. What can be inherited through the germ cells is DNA and DNA variations, as well as some epigenetic variations. For example, small RNAs, but not just. We looked at soma-to-soma transmission, so how there is kind of developmental reconstruction that is happening in one generation that is leading to the same type of developmental reconstruction in the other organism, kind of developmental niche construction, if you want. We looked at transmission, we thought and looked at transmission from soma to the germline. This is when a Weissman barrier is breached. The Weissman barrier is breached. Can it happen? We know now that it can. For example, small RNAs can be transmitted from somatic cells to germ cells, in C. elegans, in plants, and it seems also in mammals, a little bit of evidence. And we looked at niche construction, at the transmission of ecological legacies, ecological niche construction. And this is, and we wrote a book called Evolution in 4 Dimensions in order to study not only these ways of transmission, but also their interactions of the different systems of inheritance, as we defined inheritance defined very broadly. So there was a book called Animal Traditions, which studied this kind of thing. There are all kinds of nice pictures here I will not go into. And there is the system of symbolic language. This is La lingua della camaleonta, the language of the chameleon. Which is very, very strong in humans, and it's one of the strongest inheritance systems and most influential in human evolution, and leads to cultural evolution, also leads to this unfortunate situation. And I would like to thank you very much for your attention. And this is a tardigrade, the one that will remain after we're all gone.